Good morning. Welcome to the Harper Center on day 2 of the annual science conference. I'm Manuel Baranja. I'm the chair of the science committee of ICES, and it is my great pleasure to introduce today's keynote speaker, Dr. Richard Feely. Richard is a senior fellow at the NOAA Pacific Marine Environmental Laboratory in Seattle. He's also a professor at the University of Washington School of Oceanography. He has degrees in chemistry and chemical oceanography. Richard is one of the world's leaders in the ecological and socioeconomic effects of ocean acidification on marine ecosystems. Through his role in the US Climate Variability and Research Program, Dr. Feely has been instrumental in supporting an integrated approach to global observations for carbon, hydrographic, and tracer measurements. He is also a member of the steering committee of the International Ocean Carbon Coordination Project, the goal of which is to promote the development of a global network of ocean carbon observations. Richard is a prolific author. He has more than 230 peer-reviewed publications and has been recognized worldwide. In 2006, he was awarded the US Department of Commerce Gold Medal for his research on ocean acidification, and in 2010, he received the prestigious Heinz Award for his research on global change. Richard is passionate about the topic of ocean acidification and is one of the leaders in the world in knowing the consequences and understanding the impacts of OA. It's my pleasure, therefore, to invite Richard to speak to us. Thank you.
Well, it's really a pleasure to be here today. I want to thank the conveners of this meeting to invite me to come and talk about what I consider one of the most significant new scientific issues of our day in terms of ocean acidification. The scientists have been studying this for actually over 50 years, but it's only in the last 14 or 15 years where we've really understood some of its economic and ecological consequences, and that's why I'm here today. So I'll talk about what ocean acidification is, how it works, some of the chemistries, why our coastal regions are particularly vulnerable due to multiple stressors and processes that combine to enhance acidification, and present some of the ideas that we have about future implications. Now, just what is ocean acidification? Since the beginning of the Industrial Revolution, mankind has released 2 trillion tons of carbon dioxide into the atmosphere, and about 28% of that has been absorbed by the global oceans. That's about 550 billion tons of carbon dioxide that have been taken up by the oceans since by the release of fossil fuel, coal, burning of coal and oil and natural gas released in the atmosphere and being absorbed at the surface of the ocean. This creates an acid. Carbon dioxide reacts with seawater to form carbonic acid, which is a weak acid. Everyone here knows what it is because it's in your soda waters, and it gives you that fizzy taste of the soda waters. We drink it every day. This is a very nice slide. I'll show the movie in a second. This is the Earth System Research Laboratory's Global Observing System for Atmospheric CO2. And the movie is going to show the changes in the CO2 levels of the atmosphere. On the left-hand side is the South Pole. On the right-hand side is the North Pole. And these are the global mean averages of CO2. If we show the movie, please. This is the breathing of the Earth with the uptake of carbon dioxide in the summertime, and it's released due to respiration in the wintertime. This short record of 16 years shows the increase in carbon dioxide throughout the atmosphere with a really increase on the order of about 2 parts per million per year. And this is on the increase. The increase of anthrogenic carbon dioxide is about 3.2%. At present. So we're increasing the rate of increase of carbon dioxide, and we can see this clearly with the baseline increase of CO2 in the atmosphere. Here's the Mauna Loa record that I just showed you here. Here is the Mauna Loa record started by Dave Keeling, started in 1957 in the red, and increasing throughout time. This is now supported by the NOAA Israel Laboratory. And the blue is the Station Aloha time series of surface ocean PCO2 measurements commensurate with that atmospheric increase. And you can see that the surface ocean CO2 is increasing at the same rate as the atmospheric increase. We've done this at several sites throughout the world ocean, and we get the same rate of increase. And here is the pH measurements at that location. So we know from measurements alone that we are acidifying the ocean. Here's the decrease of pH. It's about 0.02 pH units per decade that we're decreasing the pH. We have this at many sites throughout the world ocean. So from observations alone, we can see the acidification taking place. This is the reaction for CO2 and water to form carbonic acid. Carbonic acid is the weak acid, so it dissociates to form a hydrogen ion. That hydrogen ion is what gives us our acidity. 99.9% of those hydrogen ions immediately react with a carbonate ion, reducing the carbonate ion concentration and forming bicarbonate. And what we see then is the pH in the pre-industrial at an average surface value of about 8.2, now decreasing by 0.1 pH units to 8.1. That's a 30% increase in the hydrogen ion concentration since the beginning of the Industrial Revolution. The carbonate ion concentration has also decreased by about 16% since the beginning of the Industrial Revolution. By the end of this century, using the IPCC business as usual scenario, the A2 scenario, we will see another 0.3 to 0.4 pH unit change, which means that the acidity of the ocean, the hydrogen concentration of the ocean, will have increased by 100 to 150% on average in surface waters. And that our carbonate ion concentration will have decreased by 50%. So what we know for certain is that approximately 28% of the CO2 generated by humans has been taken up by the ocean since the mid-1700s. The ocean acidity has increased 30%. The ocean acidity is projected to increase 100 to 150% by the end of this century. And the current rate of acidification is 10 times faster than anything we have seen over the last 50 million years, and perhaps over the last 300 million years. Now, one of the statements that you will hear from deniers is that the oceans will never become acidic, so calling ocean acidification is alarmist. I would like to point out to you that acidification is the process, the direction that we're headed towards acidic conditions. It is the process that we're discussing. Think of it this way. In your body, the pH of your blood is about 7.34 or 7.35. If you move towards acidic conditions by 0.2 units to 7.15, which is still basic, you will go into a coma. That's how sensitive your body is to changes in pH. It's very similar for organisms in the sea. Very, very different changes, even though we're moving towards acidic conditions. You might think of it this way. Here in Iceland in the wintertime, the temperature is really low, about 40 below sometimes. When it goes to above zero, you think it's warming, even though it's cold. And this is the same way with acidification. When you're moving in the direction of acidification, that's the name we give it, acidification. Now, the rate of acidification is shown here on the left-hand side. The CO2 increases have increased anywhere from 10 to 150 times faster than anything we have seen in the last million years, as well as the increase in temperature over that same range. So mankind is causing a rate of change that the organisms need to either adapt to or change by evolution or perhaps die. We don't know. Now, a major component of this acidification process is the carbonate saturation. And this is the calcium concentration plus the carbonate forms calcium carbonate. So animals that produce calcium carbonate shells must have carbonate to do so. So when the acidification process takes place, we're consuming this carbonate ion and slowing down that calcification process. Geochemists have a term for that, which is the saturation state, which is this calcium concentration times the carbonate concentration divided by the apparent solubility product. The apparent solubility product increases with increasing pressure and decreasing temperature. So in surface waters, warm waters, the saturation state is high and precipitation can occur. That's why we have precipitation of shells in warm surface waters. In cold waters or in waters of high pressure, the saturation state gets below 1 and the calcium carbonate shell or skeleton begins to dissolve. This is true for some organisms while they're still alive. So it's my job, what I do is find out where this equilibrium place is in the oceans and find out how anthropogenic CO2 is changing the location of that equilibrium point. The common carbonate minerals are aragonite, pteropods, corals, which are more soluble, and calcite coccolithophorids foraminifera, which are about 50% less soluble. Here is a plot of the changes in atmospheric CO2 due to carbon release. Here's the changes. Here's the amount of carbon that may be released. Here is a thought study done by Yost et al. Showing what would happen if we stopped CO2 emissions in 2000. Here's the atmospheric CO2 that would occur. We would still increase CO2 up to about 400 microatmospheres, and they would then slowly decrease. If we have CO2 emissions that go out to about 400 or 500 parts per million CO2, or close to 850 CO2, you can see the very, very slow change by the uptake in the oceans. It will take over 10,000 years for the CO2 to get back to pre-industrial levels if we have CO2 emissions of 2,000 petagrams of carbon, releasing 850 parts per million in the atmosphere in the IPCC business as usual scenario. Here's the corresponding changes in aragonite saturation for the surface oceans. You can see we would get down to from the normal level of average saturation state for aragonite, about 3.4, all the way down to perhaps as low as about 1.8. This would have major dramatic impacts on the ability of corals and other calcifying organisms to produce their shells or skeletons. So in order to observe these changes that are taking place, an international effort has developed to develop a global ocean acidification observing network. Right now we have about 32 moorings out in the global oceans. We're hoping to get as many as 60 in the global oceans. So we have many that are being deployed and many that are under planning. And we're working together internationally through the International Coordination Center for Ocean Acidification to develop this observing system of ships, of repeat hydrographic cruises, of moorings, of floats and gliders, all designed to work with the International Carbon Program and the International CLIVAR Program to observe the chemical changes throughout the world oceans. And I'm proud to say our newest contribution is here just north of Iceland. Dr. Jan Olafsson is directing that effort, and we've just deployed the first MAP CO2 system in the Arctic Ocean, and it's operating now, and we're getting excellent data. Thank you, Jan Olafsson, for helping working with us to put that system out. What we have done in the period of the 1990s is to determine the amount of anthropogenic CO2 in the Atlantic Ocean, in the Pacific Ocean, and the Indian Ocean. You can see most of the anthropogenic CO2 still resides in the upper 1,000 meters of the water column. This is because as CO2 is exchanged across the air-sea interface, it penetrates slowly into the ocean. And in the high latitude regions of the Arctic Ocean and the Antarctic Ocean, it penetrates slowly to the bottom and then moves along the bottom to the interior of the ocean. And it hasn't done that yet. That whole process takes over 1,000 years to occur. So most of the anthropogenic CO2 use, you can see, is still in the upper 1,000 meters of water column, where it can impact our marine organisms. And that saturation state, where aragonite will go to undersaturation and dissolution will occur, is quite a bit different. It is very, very deep in the North Atlantic. And as that water moves from the North Atlantic to the south, to the Indian, to the Pacific, it takes up carbon dioxide from respiration processes and accumulates that in the water column, such that in the North Pacific, that aragonite saturation horizon is very shallow. It's about 200 meters in the North Pacific, and in the Arctic Ocean, it's even shallower yet because more carbon dioxide is taken up in colder waters than warmer waters. And so the fastest acidification takes place is in the high Arctic region and the high Antarctic region. And what this next slide is a very important slide. It shows our natural saturation state for seawater in the 1800s based on a model, the NCAR CCM3 model. And you can see that in the tropical regions, saturation states are quite high, around 3 or 4. But in the high Arctic regions, they're already quite low, it's right around 2 or so. And you can see then the Arctic and the Antarctic region are poised to go undersaturated fairly soon. Now we're going to run this model out from 1880 to 2100. And I want to show you the changes in surface water saturation. You can run the model, please. So we can see that beginning with the uptake of anthropogenic CO2, we begin to see more red colors in the Arctic Ocean, and then a little bit later beginning to see red in the Antarctic Ocean. We can see that the tropical regions are starting to increase their saturation state. By the end of this century, you can see the entire Arctic Ocean will become undersaturated top to bottom. This will start in the year about 2034, 2035 in the Arctic Ocean. The Antarctic Ocean will also become undersaturated from top to bottom, starting a little bit later, around 2040 or so. And the entire tropical region will have greatly reduced saturation state. This is using the business as usual scenario for anthropogenic CO2 uptake. In our coastal regions, we have to think of other processes that will combine with the natural uptake of anthropogenic CO2 that can enhance the acidification process. Along the Washington, Oregon, California coast, for example, we have a natural upwelling process that takes that water that's deep in the water column that is already enriched in CO2 with respiration products, including the anthropogenic CO2, and that water gets upwelled along the bottom all the way to the beach and then gets pushed out by the surface waters that are being pushed offshore when the winds shift to the northwest. And this upwelling process brings in a concentrated level of CO2-enriched low pH waters to the beach where all the organisms are existing and then spreads out from there. We can see this process. So this is very important because this points out that in this process, the CO2 levels in the bottom waters are many times higher than that in the surface waters. If the surface waters at a present day CO2 level of about 400 parts per million, the bottom waters may be as high as 1,200 parts per million CO2. And those pHs may be as low as 7.7 or 7.8 right now. This gets upwelled and brought to the surface at the beach and then gets pushed out. So when you're thinking about acidification process in our coastal waters, you have to think of these combined impacts of respiration that's occurring in the bottom waters and that movement of those waters towards the beach. A very important process. Many of our organisms are already seeing very, very high CO2, low pH waters right now. This is a model output. I want you to focus your eye on the pH of seawater. The very low pHs are about 7.8 or so. The high pHs are about 8.1. And this starts on January 1. The upwelling process starts in April. I want to show you, start the movie, I want to show you the changes that take place. So look for the blue colors. You can see starting around day 90, you will begin to see the blue colors coming in. You see the upwelling along our entire coastline. By midsummer, we have enhanced upwelling. You can see that it goes to the beach and then gets pushed out into the coastal waters from the beach outward. This is the normal process of the enhancement of the acidification. The anthrogenic CO2 signal on this is only 20% of their overall CO2 pH changes. The rest of the signal is due to respiration processes from upwelling and local respiration processes. In this region, 30% of the water column is undersaturated with respect to aragonite in the upwelling season in the summertime. In the pre-industrial time, it was less than 10%. So we're seeing major changes in this region because of the enhanced CO2. By 2050, 70% of this water column will be undersaturated with respect to acidification. These changes are coming at us very, very fast, and that means we have to develop adaptation strategies to counteract these processes very, very fast. So experimentally, we do experiments in our laboratories. Now we have many laboratories throughout the world. We do these kinds of experiments with single organisms, changing CO2 levels. We also have mesocosm experiments, fairly large mesocosm. Some of the most best mesocosm experiments have been done here in Europe through the UPOCA program. And you can actually put systems over coral reef systems and study the changes in the laboratory and in the field. And we do this quite often. Here's an example of the UPOCA experiment that was done at Svalbard in 2010 with 9 different mesocosm experiments at levels of CO2 from 180 to over 1,000. And the results from that have become very interesting over time to see how much we can understand about the basic trophic levels. One of the things we've learned from the Svalbard experiment is by the addition of nutrients into the experiment, we can see the changes that will take place. Strongly enhancement of some of the cyanobacteria because of being stimulated by the high CO2 when there's plenty of nutrients around. We can see moderate stimulation of some of the nanophytoplankton, but it turns out that the nanophytoplankton seem to outcompete the diatoms at high CO2 levels. This suggests that we might see dramatic changes in the ecosystem at these different levels with these different species of bacteria and phytoplankton. And this would likely have significant changes in the food web as we go through it. So when we think of ocean acidification, we got to remember that CO2 concentrations will go up, bicarbonate will go up, carbonate will decrease, pH will decrease. This will affect photosynthesis, as I've just shown, decreases in calcification, changes in physiology. Also, we're beginning to see changes in animal behavior. Other changes that will occur at the same time, temperature will go up, oxygen will go down, so we need to begin to understand what the changes that will be occurring in the future under these multiple stressors that will take place globally. In addition to that, we have regional stressors of overfishing, pollution, oil spills. It's going to be a difficult problem. We have much to learn about how to determine all these different stresses. The challenges of multiple strategies over the next century will be immense. For calcification processes, we must think about a variety of different issues that it impacts. First, it will impact food security. Many of the calcifiers that we like to have available to us are people food. And in fact, in the United States alone, about 50% of the fish production are calcifying organisms. So this is a big issue for us. Fish also like to eat calcifiers for food. In fact, about 50% of all fish feed on calcifiers for their food. So this would mean that there would be an ecosystem change. And the structures that calcifiers provide ecosystem structures, such as coral reefs and clam reefs and other kinds of ecosystem structures that provide rookeries for many of our fish and other ecosystem animals. In the Pacific Northwest in 2006 and 2007, we had a major oyster emergency that took place. We have 5 hatcheries. Those 5 hatcheries in the Pacific Northwest provide all the oyster seed for the shellfish farms in the United States. And 4 out of the 5 oyster hatcheries began to lose production of the oyster seed. These are the little oyster seed. Oyster seed production went from 100% down to 20% in as much as 2 years. And this was a major implication for our shellfish industry because in the oyster industry alone on the Pacific Northwest is a $110 million industry involving 3,200 jobs. What we found was that the cause of the destruction of the oyster larvae in the shellfish oyster hatcheries was the upwelling of this reduced pH waters, which were getting into the hatcheries and causing the death of the oyster larvae within 2 days. When they would respawn the organisms, they would again cause the deaths within 2 or 3 days. So what we did is we got some resources from the federal government, and working with scientists, we put the actual CO2 sensors that we have on the ships right into the tanks in the hatcheries. And this is the first results from that. And this was a study that was done in the tanks themselves. And at decreasing saturation states, we see a decrease in larval production to the point of 0 at a saturation state of 1.6. This is the very first tipping point for ocean acidification that has ever been observed in a commercial hatchery. And by the time the saturation state got down to 1, all the animals were dead. So here's a clear indication of a process directly related to ocean acidification. Now, the way this works with this particular species of oyster is that when the oyster larvae are hatched, they have an egg yolk. And they have just a few days to produce a shell. They have to get to a size of about 170 microns and produce a shell. They use the energy reserves from their egg yolk to produce the shell. So if the pH of the water is low enough, they use up all the energy in their egg yolk and they simply die. It's a very, very simple process, very, very clear process. It's not so clear for other species, so we have to understand all the individual impacts that occur in each of the different species of oysters. The impacts are large in other oyster species, but they're different at different stages. And in some cases, the stages actually magnify from one stage to the next. So we're still learning quite a bit about this. Another example of direct effects of acidification is on pteropods. Pteropods are marine snails that swim in the water column. They have a beautiful translucent shell. This is what they look like. Their foot of the pteropod has been developed into wings, and they swim like butterflies. If you show the movie, we'll show you this is a pteropod. They're usually on the order of about 1 to 2 millimeters in diameter. This is a pteropod. This is Limacine, Helicine. They live at depth in the water column, about 100 meters or so. They come to the surface at night to feed. They throw out their nets and gather phytoplankton, and they feed on phytoplankton. This is an experiment with pteropods in acidified water at 1,100 microatmospheres for 29 days, and you can see total dissolution of the pteropod shell within about 3 weeks. This is some recent work by Nina Benarsek, who was with the British Antarctic Survey. She conducted some research in the Antarctic regions of the South Atlantic, showing us that pteropod dissolution is occurring right now when upwelled waters comes in contact with that anthrogenic CO2, and we see extensive dissolution of pteropod shells in the natural environment. We have replicated these experiments in the U.S. coastal waters and see similar types of very dramatic impacts. Pteropods are a very important component of the marine food web because they provide a very significant food source for first-year salmon and mackerel and other species. And so what happens to the pteropods is very, very important throughout the ecosystem. And in fact, some of the modeling work that has been done by the fisheries scientists in Northwest Fisheries Science Center has shown that perhaps 45% of the first year's pink salmon, 45% of their diet is pteropods. And a 10% decrease in pteropod production could cause as much as a 20% drop in the mature adult weight of a pink salmon. So here's a good example then of a food web transfer process. Now, when we look at the seafood industry within the United States, it's about a $4 billion industry throughout. Half that industry is calcifying organisms and crustaceans. The other half is finfish, and about half of that, they eat shellfish for their livelihoods. You can see the significance for our economic system of understanding acidification and how it affects the food web. So what other impacts do we see on marine ecosystems? Here's a very nice study that was done by Katerina Fabricius and colleagues in Milne Bay in Indonesia, in which they came to a region that was a very lush coral reef system, and they found some venting CO2 systems in this coral reef system. This was a natural release of pure CO2 from volcanic processes within this water. These are CO2 bubbles being released from the seafloor. And going from this very plush ecosystem to the center of the ecosystem, we saw a 90% drop in the different species of coral reef organisms. Having pH conditions, they're what we expect to see throughout the world oceans by the end of this century. So these kinds of studies in natural environment give us a clue of the kinds of changes that might expect to see. We saw a very dramatic drop-off on calcifying organisms at all taxa, as well as an enhancement of some seagrasses. So you could see there would be some shifting in the ecosystem throughout the process. For marine ecosystems, about 11% of the phytoplankton are coccolithophorids, which are marine plants that produce calcium carbonate shells. They're eaten by copepods and pteropods, and it's the copepods and pteropods that are eaten by the fish. So you can see we would have a food web transfer process that may be significantly changed in the future. If we look at the metadata analysis of what the changes that have taken place so far in terms of experimental results showing negative impacts, which are shown here in red, no impact as shown here in yellow, and positive impact shown here in green, over concentration levels from about 500 parts per million all the way out to 10,000 parts per million. What we find is very, very significant impacts with corals and echinoderms, strong impacts with mollusks, lesser impacts with crustaceans. They seem to be more resilient. But surprisingly, many of our experiments with fish larvae show some very dramatic impacts as well. So the early studies are showing from various groups very significant impacts that we need to have a better understanding of. You must realize that we're only still measuring a very small number of animals in each of these groups, so we have a long way to go. But these kinds of meta-studies give us a very clear picture that many groups of organisms will be impacted in the future. This is a list of the different kinds of processes that are affected. I've talked about calcification, shifts in nutrient and trace element speciations. pH is a master variable for the chemistry of seawater. It affects the speciation of many of our elements and compounds in seawater, particularly some of our toxic trace metals and other organics and compounds, including nutrients themselves. They're pH dependent, their speciation. So we may expect significant impacts that would affect shifts in phytoplankton diversity and ecology, effects on growth and development. Many times we see that the larval stages of the organisms are much more sensitive than the adult stages, and the reduced tolerance to other tracers. Some of the most interesting research has been done on the combined impacts of acidification, increases in temperature, combined impacts on coral species, in which the combined impact is much more severe than the two impacts by themselves. Changes to fitness and survival, changes to biogeography. We might expect some organisms to be squeezed out of their habitat and other organisms taking their place. changes to food webs. Acidification affects sound absorption, affects the ability of organisms to find their homes, their navigation ability. And some of the most recent effects, behavioral effects, have been quite interesting. A lot of interesting work in Australia that shows that for example, clownfish, their ability to avoid being eaten by predators is dependent on the CO2 levels in their blood, because the CO2 levels affect the neuroreceptors, and the responses are very dramatically. At high CO2 levels, they swim towards their predators instead of away their predators, and are quickly eaten. When you put these organisms back in the wild, they get eaten quite quickly. There's impacts on recruitment. Just the stickiness of clams and oysters and mussels to the bottom is highly pH dependent. What we know from the research right now is really quite small yet. We have a long way to go. Most of the experiments have been with abrupt perturbation experiments, usually with single species or strains. Under short duration, very few experiments have been under long duration. That's changing now. And often with very extreme pH changes. We haven't seen much work yet on the subtle changes. We have a long way to go. We need to understand the responses of genetically diverse populations, how their gene pools will help them to recover from these processes, the synergistic effects with other chemical and tracers, physiological and microevolutionary responses, species replacements, ecosystem responses, and impacts on climate change. The changes in the carbonate system are so important in the oceans. They actually provide a feedback mechanism to the atmosphere and have an impact on climate change. And we're just beginning to understand some of those potential feedback mechanisms. Now, there have been some significantly new research in the last 2 or 3 years that has shown that we have some hope that if you begin to study adaptation processes using experimental approaches, we can see that there are potentials for adaptation in some species. And this is really quite new and exciting. Here's some examples that have taken some very nice work done in Germany, showing that if you run 500 generations of coccolithophorids through 500 generations, there are some indications they can be rescued. The impacts that they are seen in the very early stages seem to be recovered from through genetic changes over time. Similarly, some breeding experiments have shown that the ability for organisms to defend themselves by changing their genetic structures, and this can be inherited from one generation to next. And probably the most significant new finding is that when you look in the gene pools and in the new technologies for genetics, looking particularly at proteins that affect many of the vital processes throughout the organisms, we can see that even when you don't see changes in the physiology, there are significant major changes in the protein structures that are ongoing. This study here studied 12 million different proteins in the purple sea urchin, and found over 40 different classes of proteins that were undergoing major evolutionary changes throughout the course of these very unique experiments. So we can see that there is hope. The ecosystem may be able to respond. It's our jobs as scientists to find out which species can respond and why they can respond, and which species cannot respond, and how can we develop adaptation strategies to help them along through this process. The way we do this is through observational, developing these global monitoring systems to measure the changes throughout the water column, to continue the research in the laboratory in the field to understand ecosystem responses, to begin to model these changes both from an ecosystem approach and a biogeochemical approach. An ecosystem approach would take us all the way through from ecosystem responses to socioeconomic responses and economic impacts to help the shellfish growers and other groups develop adaptation strategies. In the Pacific Northwest, We developed an adaptation strategy that is quite successful by changing the pH of the water that goes into the tanks, and now their production is actually higher than before just by developing a very simple adaptation strategy. As we go through these processes, we need to develop adaptation processes that will be successful in the open ocean. And most importantly, develop a very strong education and outreach, so let people know how important this overall processes are to our economies and to our ecosystems throughout the world ocean. So in conclusion then, ocean acidification is rapidly occurring. Some local species will be dramatically impacted. We know that for certain. Biological responses are variable, and so it's very important to understand those responses to different taxa and different groups of organisms. The impacts of ocean acidification will have impacts on our food webs and on our ocean industries. Other stresses to acidification will combine with acidification to enhance the process in many, many cases. And the economic consequences of ocean acidification are real and significant and happening right now. And I want to thank you very much for your attention.
Thank you very much, Richard, for a fantastic presentation. I introduced you, saying that you were passionate about ocean acidification, and I think that you have demonstrated that in ample ways. A passion rooted in deep understanding of the process and also deep appreciation of the things that still we don't know. We have some time for questions, and I'm pretty sure there will be questions from the audience. We have a couple of microphones, so if you want to have a question for Richard, just please raise your hand and I'll pass the microphone to you, and if you're closer to that microphone, you can perhaps walk to it. It'll be easier. Any questions? Yes? Would you like to just move closer to the microphone here, or someone can pass? Well, I'll do it.
I'm not a geologist, but I have some idea that in the fossil record there may be information which can guide us into the adaptations that might take over a longer time scale. I wonder if you could comment on the fossil record from times when naturally carbon dioxide levels were higher in the world.
I'd be happy to do so. I just was ready for that. I get this question often. Let me set the stage for this. Well, this is the record of the history of the oceans. Here is the present. Here is 500 million years ago. And what I've shown here is the 5 major extinction events throughout our history. And here is the calcium concentration. Remember, calcium and carbon are particularly important. Here's the surface ocean calcite saturation. And here is the PCO2 in the atmosphere. And here's the mean surface pH. Now, here's where we're headed, starting from 1750, taking us out to the year 3000 in an IPCC business as usual scenario. So you can see the very dramatic potential, dramatic drop in pH. In fact, the drop in pH that we might expect to see could be as significant as anything we've seen in the past. This occurring over the course of just a few hundred years. Each one of these events took place over a few million years. The other thing to point out is that there have been times in the past when very, very high CO2 levels existed. But you notice that the saturation state was not that much different. And the reason for that is if the oceans are allowed to respond to an increase in CO2, they will adjust to it by reacting of CO2 with deep sea sediments, dissolving those sediments, and therefore increasing the alkalinity and hatching that saturation state. So the rate of change is very, very important to this overall process. And if our process takes a few million years to occur, the oceans can adjust. What we're doing is changing the ocean over a few hundred years, in which all that anthropogenic CO2 is still up in the surface waters. And so the oceans have not had time to adjust. During the PETM, for example, which was the last big significant event that had CO2 increases, about 4,000 gigatons of carbon were released. It took 125,000 years for the oceans to adjust back to its pre-level. So we can see major, major changes taking place. And we can see that the difference now is the rate of change, that mankind is changing it so fast, the oceans simply do not have time to adjust.
Thank you. Other questions ready for the next slide? Yes.
So I guess there's some people who argue that the cause and effect is reversed, that the fact that the temperature is increasing is leading to the release of CO2. You know, if one looks back in history and looks at leads and lags. So I guess my question is, how well do we understand the modern day budget that we can track the amount of CO2 being emitted by humans and track it through the atmosphere and ocean to sort of refute that claim or address that claim?
Well, I can say that because of the very good models that we have that exist right now, we have a fairly good understanding how the acidification process will work in the future, depending on the amount of CO2 emissions released. I showed some examples of that earlier in the presentation. If you have a business as usual scenario in which you expect a temperature increase on the order of 2 to 4 degrees C, the effect of that temperature on the acidification of the oceans is very small. It is measurable, but it's very small. The effect of CO2 enhancement of the ocean chemistry is much, much larger than the effect of the changes in temperature. And this is true throughout the world oceans. The dominant impact is the uptake of the anthropogenic CO2 by far. And the chemistry is so well understood and the models are so clear on that for the surface oceans that we're very certain about that.
Thank you. I've got one question myself, and then we'll go to you. And this, Richard, one of the consequences of the OA research has been a more regular monitoring of pH, not just in the open ocean, but in coastal zones. And what we've seen now is that there's a huge variability on pH on a daily basis and seasonal basis in very coastal region close to the rivers and the like. Would you like to comment on what are the consequences for that in terms of adaptation abilities of organisms near the shore?
That's a really great question, and there's just been a very tiny amount of research on that. I think it's going to be, again, very organism dependent. In other words, some organisms have learned to adapt to the high variability of pH in water, and some studies have shown that in regions of very high variability, the adaptation of those organisms is quite good. In regions of low variability, the adaptation of those organisms is not so good. Now, what that means for evolutionary response, we're not quite clear yet. We haven't done enough of those studies. But there is suggestions that some organisms are able to adapt. We just don't know yet how many organisms have that capability or not. One of the things that we have learned from the observations themselves is that we can see the anthrogenic CO2 signal in high variability regions if we measure it a long enough time. Along our west coast, for example, we've had time series measurements for as long as 20 years or more, and we can see that anthrogenic CO2 signal. So with observations taken long enough, we can see those changes. And the other point to make is that those changes are so dramatic that they exceed that natural mean of that and variability about every 40 years or so in our coastal water. In the open ocean environment, we exceed that envelope of variability every 20 years or so. So it's dramatically different, and we are seeing those changes now. As I said, in the Pacific Northwest region, we will go into very different chemical changes within the next 30 or 40 years. So the organisms really have to respond quickly.
Thank you. Next question.
This is a somewhat related question. I was thinking, in the coastal environment, the pH typically has a wide variance, in seasonal and annual time scales. I was curious if the actual impact on organisms is the pH itself, the actual amount of acid there, or is it on the change in carbonate or saturation constant? It's typically the most important aspect.
Again, it will be different for different organisms. In general, it's not the hydrogen concentration that is the major impactor. It appears right now, at least for calcifying organisms, it's usually carbonate ion or bicarbonate ion. Saturation state seems to be the major impactor or the major controlling factor for calcifying organisms. It's not so clear for other organisms. We're still getting a better handle on that. It depends dramatically on the metabolic processes that take place. And some of the research has been suggesting is that the tolerances of fish and other organisms to temperature at their extremes, either low or high temperatures, are greatly reduced the higher amount of CO2 in their blood systems. So you can see that we may be impacting the habitats by the physiological processes that are occurring in the blood systems itself at higher CO2 levels.
Thank you. Other questions? Okay.
There's one over here.
Oh, sorry.
Is it possible that some of the organisms, when they dissolve, that the dissolution itself will be a feedback that will then raise the pH? So I've seen some work recently, I guess with oysters, that suggests that might happen.
Yes, that's true. And there's been a number of studies that have taken place to assess how that significant that will be. And it's hard to evaluate quite yet. The questions we have, for example, in the open ocean are quite different than the questions in the coastal ocean. In the open ocean, what we're worried about is that the ballasting, the carrying of carbon from the surface ocean into the ocean interior is done by piggyback on carbonate and silicate particles. So what happens when those carbonate particles begin to dissolve at shallower and shallower depths? Well, that means that organic matter is then released, and therefore respiration will be enhanced shallower and shallower in the water column. So we would expect that to have a very dramatic feedback on climate, and in that case, enhancing the negative climate feedback. And the models on that are very rudimentary right now. They show that this is a very important process in the open ocean. We need to have better understanding of carbonate dissolution, where it occurs, how it occurs, which organisms are affecting carbonate dissolution. In the coastal ocean, it's quite a bit different issue. It's how quickly that dissolution will take place and how much alkalinity will be released back in the water column. And Andreas Andersen and Fred McKinsey have addressed that in their book. And what they have suggested that this process does exist and it will have a change in alkalinity, particularly in coastal lagoonal systems, but it's not enough to counteract the very strong impact of CO2 emissions. It's like orders of magnitude too slow for that process.
Thank you. Behind you on the foot.
Yeah, if we put this together with the opening lecture, we're reminded that common property resources get overexploited, and the oceans and the air are common property resources. So I guess the business as usual is the most likely scenario for us to see. Now, in the case of global warming, you can perhaps think of mitigation things that you might do as a plan B, as opposed to a low carbon scenario. Is there anything at all that could be done in terms of mitigation on a sane sort of level? I find it very difficult to believe that there is.
I'm not sure I understand the question. You know, what can we do on a global scale?
Well, not necessarily on a global scale, but perhaps on a local or a shelf scale. Is there any, if we are faced with your scenario on your bottom of your slide here,
Yes.
our business as usual, and that seems to me the most likely scenario given human behavior, then is there anything at all we can do, or do we just have to live with whatever the consequences are?
That's an excellent question, and if you look at many the presently existing geoengineering approaches, none of them work for acidification at all. Some of the ideas that are on the table, however, are interesting, and they are meant and designed for local adaptation, not global adaptation. For example, there's a lot of interest on adding lime to CO2 emissions, which would create alkalinity, and then releasing that slurry of alkalinity into the oceans. That's been one suggestion. Another suggestion has just been adding lime to the ocean directly. These are suggestions for abating local impacts, and the problem with them is they have only been studied in laboratory settings, so we don't know what indirect impacts any of these approaches would have right now. And there's been lots of interest in trying to enhance some of these studies to see if you can see scaling up a little bit what the impacts might be. The costs are prohibitive, so they can't be done on a global scale. And it may be helpful, though, for some local environments, like, for example, shellfish environments. And that's what's being addressed at present. Again, most of the experiments are only in the laboratory at present, and very poorly funded.
Thank you. We might have time for one more question before coffee. If not, yes. Oh, now three. Okay. Well, I'll take this too.
I wonder if you could say a few words about the art of measurement of CO2 and pH. Is that a technology that's well in hand now, or is there a need for improvements? Are there uses for expendable types of probes that could be used on ships of opportunity and so forth?
That's a wonderful question, and I really appreciate you bringing that up. We're at a stage right now where we can make very precise and accurate pH and carbonate system measurements on board ship with the same kind of instruments we would have in our laboratory on board ship. We can measure pH to an uncertainty of about 0.0015 on board ship. We can measure total CO2 to uncertainty about 2 micromoles, and alkalinity about 2 micromoles. It's quite a bit different when we try and put sensors on moorings and on floats and gliders. We are working quite hard to develop sensor technology, and there have been some very recent successes. For example, we have a new pH sensor called a Durafed sensor that measures pH on moorings, and we have placed them now on floats and gliders for the first time. And they don't measure at the precision and accuracy that the shipboard measurements make, but fairly good. They're at the level about 0.01 or 0.02, so that we can get the physiological responses and address some of that variability issue that we need so bad. It's a little harder to have them quite ready for long-term climate change, but we're getting better. And there's a lot of interest in this field right now, and I'm happy to announce that just this last month, the X Prize Foundation has put out a $2 million prize to anybody in the world that can build in a cheap, reliable, and affordable, high-accuracy pH sensor. And we think the interest in the X Prize and the process will develop a lot of development of new technology that will be available very, very quickly. So I offer this opportunity for everyone who has a good idea for a pH sensor to go for the X Prize. It's wide open.
Hi. Okay, so my question is in reference to coupled biogeochemical cycles, and basically what I'm interested in knowing is, you know, a lot of these laboratory experiments are looking at things relatively statically in terms of other chemicals and the coupling. So how will OA affect coupled biogeochemical cycles in future oceans?
You know, the models for that are actually quite simplistic at this point in time. The biogeochemical models are very, very simple models, and they don't include a lot of the more sophisticated geochemical processes that we can measure in the laboratory. And they don't include the ecosystem changes right now. For example, when we see very dramatic changes in cyanobacteria, that will affect nitrogen cycle, as you well know. And therefore, these kinds of changes in the nitrogen cycle are not embedded in the large-scale global models right yet, as well as the changes in the basic distribution of phytoplankton and zooplankton. So there are many examples like this in which the nitrogen cycle would be affected. The trace metal cycles they're not in the global biogeochemical cycle models yet for the most part. There is some exception. Iron's in large-scale models, but some of the other trace metals are not yet. So we have a long way to go into developing the sophisticated biogeochemical models yet.
Great, thank you. It's now getting to coffee time, and I'd just like to use the opportunity to again thank Richard, not just for a fantastic presentation, but an even better question and answer session, and in general for all your work on OA in science, on politicians, society at large, the public. So thank you very much, Richard. If you please show with your appreciation while I give Richard a couple of presents.
Oh, thank you very much.
To add to your luggage allowance.
Yeah.
Thank you.
Thank you very much.